Good day, everyone. My name is Dimitrov, and I am a PhD student in the group of Julio C.S. Rodriguez at Heidelberg University. And I will now very briefly present our recent comparison of methods and resources used to infer cell-cell communication from single-cell RNA-seq data. But before I start, I would like to thank everyone involved in this work and also thank the organizers of this event. So cell-cell communication is essential in multicellular organisms, and it plays a part in almost any process ranging from homeostasis to disease. So unsurprisingly, the technological advances in both dissociated and spatially resolved single-cell data have sparked an immense interest in exploring intercellular communication. And this has in turn enabled us to gain an additional layer of insights. So typically, cell-cell communication inference from single-cell data aims to predict the relevant interactions between any pair of clusters in the dataset. These interactions are usually predicted by the gene expression of a transmitter protein expressed by a cluster used as the source and a receiver protein expressed by the target cluster. The transmitter-receiver interactions between any pair of clusters are then prioritized, and each tool uses a different method to do this. So albeit different types of methods exist, including data-driven methods that take spatial information into account, or such that consider the downstream signaling. In the most common case, intercellular interactions are obtained using a resource of prior knowledge, together with a method to prioritize the interactions. And usually the method and the resource come in a fixed fashion. So to decouple the methods and the resources, we built Liana, a ligand receptor analysis framework that at its current state serves as a common interface for 15 resources and 6 methods. We then used Liana to assess how does the choice of method and resource impact the predictions. Since the methods rely on prior knowledge, the choice of resource and any potential bias in it are expected to impact the results. As such, we examined whether specific pathways and biological functions were unevenly represented in specific cell-cell communication resources when compared to the collection of all resources. So as an example, we matched transmitter-receiver interactions to pathway categories from signaling and observed the relative underrepresentation of the Wnt pathway in the guide to pharmacology and cell phone DB resources, as well as the relative enrichment of the RTK pathway and the OmniPath resource. We further investigated the relative bias of the resources when considering other functional databases, but for the sake of time, I would kindly refer you to our manuscript. So then, to systematically evaluate the relative agreement between the methods and the importance of the resources, we ran all possible combinations of methods and resources on publicly available datasets. We did this under the assumption that regardless of the method or the resource used, we should see a general agreement when using the same expression data. Yet, what we saw is that both the resource and the method had a considerable effect on the predicted interactions, and this is presented in this heat map, which simply shows whether an interaction is present or absent in the 500 highest ranked interactions for each method-resource combination when using the same colorectal cancer dataset. So the heat map further shows that the impact of the methods outweigh that of the resource as the different method-resource runs clustered by the method, and this is maybe easiest to see in by the dendrogram. So, in summary, what we saw is that the choice of both the method and the resource can considerably impact the interaction predictions. And in the manuscript, we further built on how these differences could affect the downstream interpretation of results. So, acknowledging the great contributions of all scientists involved in this field and the exploratory potential of these cell-cell communication tools, we also believe that there is a further need to better understand and evaluate them. However, this also begs the question, how do you benchmark that which has no truth, as the ground truth for cell-cell communication inference is largely lacking? Nevertheless, there are certain ideas that are already brewing in the community, and we hope that Liana will be valuable for the future benchmark of the methods. So this was my presentation, and I thank you for your attention.
Hello, everyone. My name is Bobby, and I work at the Genome Institute of Singapore. I want to thank the organizers for giving me the opportunity to present Dubstepper, a correlation-based feature selection method for clustering single-cell RNA sequencing data. Heterogeneity in single-cell RNA sequencing data is conventionally characterized by unsupervised clustering, which has a number of steps which are still being developed. We decided to focus on feature selection, that is, selecting the optimal set of genes to separate cells into distinct clusters, because it is a critical and relatively underlooked step. Current methods in the field look at genes individually. So they look at variance to mean ratios of genes, for example, the highly variable genes method, or dropout rates in the context of M3 drop. However, when we analyzed a cell line dataset from Tian et al., we found that the top marker genes of a cell type tend to be strongly correlated with each other, as demonstrated in the gene-gene correlation heat map on the right, while marker genes of different cell types tend to be negatively correlated with each other. We wondered if we could use this information and thus constructed a correlation range score, which is basically the scale difference between the strongest positive and negative correlations of a gene. Indeed, we found that DE genes, or cell type specific genes, have a significantly higher correlation range score as compared to non-DE genes. And this score is also predictive of fold change and Q value of differential expression. Thus, we use this information to build Dubstepper. The first step in Dubstepper is separating genes which have a high correlation range score from those which don't. These genes are then used to construct a gene-gene correlation matrix upon which we perform stepwise regression to identify unique patterns of expression. We use a scree plot to determine the elbow point to stop performing regression so that we identify the minimal set of representative genes, and use a guilt by association approach to add correlated genes to this feature set to fortify the directions of variation. Finally, we use a novel density index metric which measures local neighborhood density to determine the optimal stopping criterion or the optimal feature set size for a data set. We benchmarked Dubstepper against 7 existing feature selection methods on 7 data sets with objective ground truth, that is cell lines or fact-sorted data sets spanning 4 scRNA-seq protocols. We quantified performance using silhouette index, which measures cluster separation, and AUROC, or area under the ROC curve, for marker gene detection that measures sensitivity and specificity of selecting marker genes in the feature set. In terms of both silhouette index and AUROC, we found that Dubstepper does outperform the existing methods. However, the second best performing method in the case of silhouette index is the high loading genes method based on PCA, while in the context of DE gene detection, it's the new state-of-the-art deviance-based feature selection. Density index that I introduced earlier allows us to optimize this feature set, and in 5 out of the 7 data sets that we analyzed, provides an improvement in the silhouette index over the peak that we saw in the previous result. Finally, we can also generalize Dubstepper to SC-ATAC-seq data because it's based on correlations, which can be extended from genes to peaks. We compared Dubstepper against the default, which is no feature selection in the field, and an extension of highly variable genes to peaks, which we call highly variable features. And indeed, we find that Dubstepper is able to recapture the differentiation schematic of hematopoietic bone marrow cells much better than the highly variable features or the default in the field. In conclusion, we wanted to prove using Dubstepper that pairwise feature correlations contain important information for selecting cell type specific features. We showed that the density index provided by Dubstepper can be used as an optimal stopping criterion for feature selection. Further, Dubstepper is available as an R package on CRAN, and its source code is on GitHub. I would like to acknowledge the lab, especially my PI Shyam, for his support over the last couple of years, funding from GIS and ISCB, and I thank you for your time. Please check out my poster in the poster session and the preprint for Dubstepper on bioarchive.
Hi everyone, I'm Marius. I'm a PhD student in the lab of Fabian Theiss in Munich, and I'll be talking about cell rank for directed single-cell fate mapping. So the starting point of cell rank was the classical concept of trajectory inference in the single-cell field, where you basically derive a robust distance metric on a graph that lets you infer continuous biological trends from snapshot data as you get it in a single-cell RNA sequencing experiment. So this has been tremendously successful in the field and works well in many situations if you know the starting point of the process. So here's an example of the developing pancreas. If you give this population to the algorithm as a starting point, it will give you the correct temporal ordering. However, under perturbed situations, think of regeneration, reprogramming, etc. This is much more difficult because you often don't know where the development starts and where it ends and what the direction in between should be. So in this situation, Rna velocity comes into play. This famous paper by Lomano et al. From 2018 that basically gives you something like an estimate for the first derivative of gene expression. So it can tell you whether a gene is currently being up or down regulated. This is actually a high dimensional measurement. So you get this for every single gene. This has been extended by my colleague Volker here on the right in his SCVelo package, which extends the original idea also to transient populations. Now, this is extremely powerful, and what has mostly been done with this data, this Rna velocity data, is that it's projected into low dimensions to see the main developmental trends, which is great. However, we argue that for detailed questions of trajectory inference, you should actually work directly with the high dimensional data. So with the high dimensional velocities that you get per gene. Now, this is where cell rank comes into play. In cell rank, we're really trying to combine the best of both worlds. We're trying to take the robustness of trajectory inference based on cell-cell similarity and combine that with the directionality given by Rna velocity in high dimensions. So we want to make use of the full high-dimensional vector field to identify both the initial and the terminal states and to assign fate probabilities. And this project is a collaboration with the lab of Dana Peer in New York. And there are many more people involved, but two other people I want to highlight here, Michael and Philip, who have been involved a lot on the software side of this. So to give you a high-level overview, we make use of an algorithm for this called generalized Barron cluster analysis. It was developed by Bernhard here on the top, which basically derives macro states of the developmental process, which can be either initial, intermediate, or terminal. And the algorithm can also help us to classify these different macro states. And once we have used this to identify the terminal macro states in this biological process, we can then compute fate probabilities towards these. We validated this on some in vitro data reprogramming. There are two states, successful, dead end. Actually, successful is quite an unlikely state here. Most cells go to the dead end cluster. There's sort of ground truth available for this data because there is lineage tracing through the cell tagging method. So when we computed velocities using either Velocite or SCVelo, when we project them into the embedding, we cannot see the path that leads to the successful state because it is overwhelmed by the signal that goes to the dead end stand. However, when we use cell rank, first of all, we identify the successful end state. And when we compute fate probabilities towards this end state, they correlate very nicely here on the right with the ground truth that you get from the cell tag labels. Next, we went to an in vivo application, went to the pancreas that I showed you before, computed the velocities, used cell rank to get the macro states, and then identified the initial and the terminal macro states, which in this system correspond to hormone-producing populations. We then computed the fate probabilities and correctly identified that beta is the dominant state here. Then we were able to compute gene expression trends of the main drivers towards these respective end states. There's another application we have in the paper where we show that cell rank really generalizes beyond normal development to perturbed conditions. So we demonstrate this on lung regeneration. However, I won't go into this. The one other thing I do want to notice is that we have meanwhile been working on extending cell rank to situations where Rna velocity is not available by modularizing it. So there are different kernels. Each kernel takes in some data and computes a transition matrix. So we have one kernel, for example, called the pseudotime kernel, and there are some more kernels that you can find when you visit the website cellrank.org. This also brings me to the end of my talk. I would like to thank everyone that was involved in this project and you for your attention. Thank you.
I'm Vivian Lee, an assistant professor at Rutgers University, Department of Biostatistics and Epidemiology. I would like to introduce our recently developed method named MAPPER, which is for model-based analysis of alternative polyadenylation using 3' end linked reads. This is joint work with the Bingtian's lab at the Wistar Institute. The polyadenylation refers to the addition of a poly A tail to an mRNA transcript, and APA is a widespread mechanism of gene regulation that generates distinct 3' end in mRNA transcripts. And APA occurring in the 3' untranslated regions can lead to the production of mRNA isoforms with different stability and can also affect mRNA translation and localization. In human genome, APA occurs in over 70% of the protein coding genes, and it has been recognized as a critical regulator of gene expression. In addition, APA profiles are frequently altered in immunological and neurological diseases, as well as in cancers. Therefore, it is a critical task to analyze and summarize APA profiles from RNA-seq data. As many single-cell RNA sequencing technologies generate RNA reads from the 3' end of the RNA molecules, such data provide a nice opportunity for studying alternative polyadenylation. In particular, we focus on technologies that generate the so-called 3' end linked reads. In this scenario, the cDNAs are generated from the full-length mRNA transcripts and then fragmented. Next, only the fragments correspond to the 3' ends of the mRNA transcripts are retained for sequencing. However, in the sequencing step, the reads are generated from the 5' ends of the cDNA fragments. After being mapped to the reference genome, most 3' end linked reads have a distance to the poly A site. As their distances to the poly A site is random, the 3' end linked reads do not provide non-ambiguous information about poly A site positions, and we need to predict the poly A sites using a statistical method. Example technologies that can generate 3' end linked reads include 10x genomics, DropSeq, and also bulk sequencing methods such as QuantSeq forward and PetSeq. This is a diagram showing the major steps of our MAPPER method. It takes the mapped reads from 2 sample groups and the poly A site database as the input, and it has 2 stages of modeling. The first stage is to predict poly A site positions using a likelihood model, and the second stage is to perform APA analysis based on statistical testing to evaluate which genes have differential APA events between the sample groups. In the interest of time, I will show one application of MAPPER to single-cell RNA-seq data. We applied MAPPER to 10x genomics data from 5 placental samples and focused on 3 trophoblast cell types, which are specialized cells of the placenta that play an important role in embryo implantation and interaction with the maternal uterus. And because VCTs are the precursor cells of the other 2 subtypes, we compared the APA profiles of EVTs of SCTs with those of VCTs. MAPPER on average identified around 13,000 expressed genes in the 3 trophoblast cell populations, among which 44% had a single poly A site. And we find that around 75% poly A sites were in the 3' most exon, and 21% were in the introns. Using FDR-adjusted p-value equal to 0.01 as the cutoff, MAPPER detected significant poly A site changes in around 5,800 genes between EVTs and VCTs and 4,800 genes between SCTs and VCTs. The RED is a score we defined based on poly A site quantification, and a positive score indicates RNA transcript lengthening, and a negative score indicates shortening. Interestingly, the genes with significant APA changes in EVTs or SCTs did not overlap substantially, indicating distinct APA regulation in 2 cell types. For example, MAPPER predicted 3 poly A site in the 3' UTR of MUC15. We find that this gene is not only differentially expressed between the 3 cell types, but also displayed 3' UTR shortening in the differentiation of SCTs. The mapper method has been implemented as an R package available at both CRAN and GitHub. If you have any questions about our method or software, please feel free to contact me for more information. Thank you very much.